So today we are going to talk about protein digestion and absorption. Now, so our topic is actually protein digestion and absorption. Now, most important point you need to understand is this, that these are actually two really large topics. These two are actually not a single topic, but these are really two large topics because they contain many enzymes, and enzymes are proteins, and proteochemistry is actually really tough. Now, first point you need to understand is this: we will talk about protein digestion, then we will talk about protein absorption. Now, important point you need to understand is this: we will be talking about protein digestion in actually two main phases. Okay, protein digestion have two main phases. Okay, that is digestion in stomach and digestion in small intestine. Okay, now first point you need to understand is this: digestion in stomach and digestion in small intestine, they are actually completely different. Why? This is because of the fact that digestion in stomach actually occurs with the help of one enzyme only, that is pepsin. Right? It occurs with the help of pepsin. We will go into details, don't worry. But digestion in small intestine is actually occurring because of many enzymes. Number one, trypsin, that you all know very well. Trypsin. Number two, chymotrypsin. Okay. Number three, enterokinase. Number four, carboxypeptidase A, carboxypeptidase B, many things. But the most striking feature about the digestion of small intestine, or digestion in small intestine, and not of small intestine, digestion in small intestine and digestion in stomach is that the digestion in stomach occurs with the help of pepsin, which is an endopeptidase. Okay, which is an endopeptidase. Okay, and digestion in stomach occurs with the help of endopeptidase and exopeptidase. Okay, that means digestion in stomach occur with the help of pepsin, which is an endopeptidase entirely. Now, first thing you need to understand is this, that digestion in small intestine occurs with the help of endopeptidase as well as exopeptidase. We'll talk about that which one is the endopeptidase and exopeptidase in details when we'll talking about this. Now, why I am talking about these things, that this is an endopeptidase, exopeptidase, this is because of the fact that from the first you must have this idea in your mind that because digestion in stomach is occurring because of an endopeptidase, you must understand that endopeptidase, suppose this is a protein, this is the N terminal, this is the C terminal, suppose this is a protein, and endopeptidase means what? It will cut this protein from inside of the protein. Suppose this is a protein, this is its N terminal, then this is its C terminal, and suppose this protein is being attacked by an exopeptidase. Now the first thing you need to understand is this: suppose these are amino acids, these are the constituent amino acids which is making up the protein. Now first point you need to understand, that is, whenever, suppose this is protein number 1 and this is protein number 2. Now, whenever you will see that protein number 1 is being attacked by an endopeptidase, that means that this protein or this endopeptidase or endopeptidases actually attack the peptide bonds which is inside the protein. Clear? The first point you need to understand is this, that whenever endopeptidases attack a protein, they attack the inside peptide bonds. That means they will, they will cleave the inside peptide bonds. Clear? The peptide bonds which are inside the what? Proteins. Now, first thing, now again, let us talk about exopeptides. Suppose this second protein is, attacked by an exopeptidase. Now, important point you need to understand is this, that exopeptidases act from one of the terminals. That means what? Suppose exopeptidases means they cannot directly act in the amino acid peptide sequences or peptide bonds which are joining two amino acids present inside the protein. That means exopeptidases cannot attack these peptide bonds which are present inside the protein. But what they can do is actually they can attack from both the ends. They can attack the peptide bonds which are nearer to N and C terminal. That means suppose if they remove this amino acid, then you are getting what? This is the amino acid, this is another amino acid. Now, suppose again they are attacking here. Suppose then you are getting what? Another amino acid, another amino acid. Now, after that, after some time, what you will get when exopeptidases are working on a protein, what you will get when exopeptidases only are working in a protein, you will get amino acids. You will get what? Amino acids, right? Because suppose this is an amino acid. This is, suppose these four of my whiteboard marker pens are amino acids. Suppose I am an endopeptidase in the first case. Then what I will do is I will actually split these proteins from inside. That is what I got, what I had earlier. I had a protein. Now when this protein is attacked by an enzyme, then we have got two segments. These are now, each segment is now called a peptide. Okay, clear? It's now called a peptide or oligopeptide. That means any protein which is attacked by an enzyme is called peptide. But that means in which case, now you tell me, in which case you will get peptides when endopeptidase is attacking a protein or exopeptidase? When endopeptidases are attacking a protein, that right? That means what? When endopeptidases are attacking a protein, you will get peptides, right? When exopeptidases are attacking a protein, you will get amino acids. You will get amino acids, right? When endopeptidases are attacking a protein, you will get peptides. When exopeptidases are attacking a protein, you will get amino acids, right? Now the first thing you need to understand is this: we as a human being absorb amino acids. First thing you need to understand. We human beings absorb amino acids. We don't absorb peptides. We don't absorb peptides, my friend. That means what I am saying is this. So digestion in stomach, is it important? Listen, many books say otherwise, but actual, scientific fact says something different. That means there was a study which was conducted on many, patients that they have gastrectomy. That means their stomach has been removed. That means no digestion of stomach, right? There will be no digestion of stomach. But still, that means what? If you don't have proper digestion of stomach, if you don't have proper digestion of protein in the stomach, then doctors thought that these proteins will end up in the blood pool and will be converted into urea and uric acid level in the blood should rise. But that didn't happen. So then and there, scientists concluded that the majority of the protein digestion actually occurs in the small intestine. Now, why it is helpful? Why digestion in stomach is helpful? Because of the fact that proteins are converted into small peptides which are easier for any enzyme to act upon. That's why digestion in stomach helps in the digestion of small intestine process. But if you actually obligate this particular step, there will be no problem. Clear? So this has been established. Now we will talk about digestion in stomach. Now listen, digestion in stomach is not important with respect to human body, but it is very much important with respect to your exams. Okay, so try to read this. Now we will talk about digestion in stomach, digestion of protein in stomach. Okay, now we will talk about digestion of protein in stomach. Now important point to understand is this, that digestion in stomach actually starts, now listen with all your attention, digestion of proteins start in the stomach when they are activated by vagus nerve. Now listen, suppose this is your stomach. Okay, suppose this is your stomach. Suppose this is your stomach. Now, important point to understand is this, your stomach have many different regions, right? This is what? This is what? This is what? Fundus, body pylorus, right? Now, important point to understand right now is this, that important point to understand right now is this, that the main cell which secretes the digestive enzyme is chief cell, right? Chief cell secretes pepsinogen, right? Now important point to understand is this, that how they secrete pepsinogen. Now suppose this is your gastric pit. Okay, suppose this is your gastric pit, and these are your, suppose, chief cells. Okay, suppose these are your chief cells. Now the first thing you need to understand is this, that the chief cells have something in peculiar with respect to pepsinogen secretion. They have some peculiar things associated with pepsinogen secretion. Let me draw it here. I'm going down and down. Okay, let me draw these things here. Now suppose this is your chief cell. Okay, so this is your chief cell which is secreting what? Which is secreting your pepsinogen. Now important point you need to understand is this, a pepsinogen is actually a zymogen. You know what is zymogen? It's a zymogen. Okay, what do you mean by zymogen? Pepsinogen is a zymogen, means what? Pepsinogen is a zymogen means that pepsinogen is an inactive precursor of the active enzyme. Clear? Now every enzyme starts with, or every enzyme ends with ase. Pepsin doesn't end with ase. Why? Because earlier when we were studying gastric physiology, pepsin was considered a protein. Actually it is a protein, don't doubt about that. It was considered only a protein, and what scientists used to do is they used to perform SDS page, and they actually got this protein with SDS page, and that's why it was named pepsin, not pepsinase. Clear? Now important point to understand is this, what is zymogen? Zymogen means it's an inactive precursor. Inactive precursor. Now inactive precursor of an active enzyme. What do you mean by this? Now let me give you an example. Bruce Wayne, Bruce Wayne is what? The main character who actually, is associated with activity in the Gotham, city. That is, his actual name is in public life, he is Batman. Now listen, Batman is the active form, but Bruce Wayne is the zymogen. Okay, Batman. Okay, Batman. Oh my God, Batman, and this is Bruce Wayne. So if you have understood that what is the role of zymogen and what is the role of pepsinogen. From pepsinogen comes pepsin, right? From pepsinogen comes pepsin. That is what? That is what is called Batman. So zymogen is actually Bruce Wayne, and with a mask, this zymogen is only called Batman, that is pepsin, right? Okay. So now you all know what is zymogen. Now listen, what I am telling is this right now, that it is a protein, right? Pepsin is a protein. That means it should have a gene. Pepsin is a protein and it should have a gene. Now first thing you need to understand is this, why we are interested in this protein, Now pepsin is a protein and it has got 3 isoforms. Clear? If you want to know what is isoform, go to the protein biochemistry section of our video library. You will understand what is isoform. Now pepsin has 3 isoforms, that is group 1, okay, group 2, and cathepsin E. Now for your better understanding, you must understand that whenever you are talking about pepsinogen or pepsin or whatever it is, you are talking about group 1. Any book which is talking about pepsinogen or pepsin, actually it will be pepsinogen. Okay, so pepsinogen has 3 isoforms, that is group 1, group 2, and group 3. Now actually what happens is, suppose this is a chromosome, this is chromosome number 6. Suppose in chromosome number 6 pepsinogen gene is present. In chromosome number 6 pepsinogen gene is present, okay, in chromosome number 6. Now important point you need to understand is this, that in chromosome number 11, but important point you need to understand is this, that in chromosome number 6 there is a peculiar type of protein called pepsinogen, okay, pepsinogen PGC, pepsinogen C. We don't know the role of this pepsinogen C, but these pepsinogen C proteins have their genes in the chromosome 6. Now let's talk about the normal chromosome, that is chromosome 11. We will otherwise forget everything. Now first thing you need to understand is this, that whenever we are talking about pepsinogen, we must understand it's a protein, right? It's a protein and it is converted into its active protein. Now important point, suppose this is your what? This is your nucleus, this is which compartment? This is ER, right? This is what? This is Golgi, right? This is trans Golgi. Now important point you need to understand is this, that these pepsinogen are co-translationally translated into they perform co-translocational translation and they are transferred to the endoplasmic reticulum. Okay, and from the endoplasmic reticulum they go to the cis Golgi, then they go to the trans Golgi, then they are thrown out of the cell with the help of vesicles. But important point you need to understand right now is this: there is a peculiarity. Why? Because pepsinogen is an active protein or an inactive protein. It's an inactive protein, right? Now it is one of those proteins which are not secreted actively outside the cell, right? That means pepsinogen from here to here goes almost without any change. Clear? Now from trans Golgi, when a protein is going out, it is called exocytosis, right? Now protein sorting. Sorting, that every protein chooses its own vesicle so that it can go to its particular target area of the cell. Now first thing you need to understand is this: when pepsinogen is going out of the cell, then they perform an very much important and intriguing part which is called compound exocytosis. They perform compound exocytosis. What do you mean by compound exocytosis? Compound exocytosis means what? That suppose this is a cell and suppose this is cis Golgi, trans Golgi, and from this trans Golgi vesicle will be produced and this vesicle will be throwing its content out of the cell. Now the first thing you need to understand right now is this, that in compound exocytosis, apart from, now listen with all your attention, in compound exocytosis, apart from pepsinogen, many other proteins are also present which are thrown out of the cell together with pepsinogen, which is very rare. Now listen with all your attention. In a neurotransmitter vesicle, will you find any other proteins? No. Will you find any other compounds associated? No, because that has been sorted. But in a pepsinogen containing vesicle, you will get other proteins together with pepsinogen. That's why it is compound exocytosis. Compound means here that together with other protein, this pepsinogen is thrown out. Clear? What now? If I tell you what is the most biochemical feature which is reflected by this particular point, you understand what I am saying? That if I tell you, or if I ask you that what is the most important biochemical feature of pepsinogen which is shown by a compound exocytosis, it shows that within the cell it is totally inactive. It never disturbs a protein. Now listen, it's very important. It's a endopeptidase. That means it can attack any protein, but when it is activated, now during compound exocytosis, it is thrown out together with what? It is thrown out together with other proteins. That means those proteins which are coming out with the help of this vesicle containing pepsinogen, they are not attacked by pepsinogen. That is very rare, right? Now if you get any questions like this, that why pepsinogens are secreted as inactive precursor, why pepsin is secreted as an inactive precursor, the answer lies there, because of compound exocytosis. Many proteins comes out together with pepsinogen, and as pepsinogens are not activated inside the cell, that means this pepsinogen actually is regulating the autodigestion of stomach. Clear? Okay. Pepsinogen activation within the stomach is really dangerous. It can autodigest the stomach. Okay. Now, so you now know what is compound exocytosis and all these things. Okay. Now, important point here you need to understand again is, suppose you have successfully thrown out you were successful, or your cells were, or your chief cells were successful in throwing out the pepsinogen. Now, important point you need to understand is this: before pepsinogen comes out into the stomach or into the gastric pit, suppose suppose this is your gastric pit. Okay, suppose this is your chief cell. Okay, before pepsinogen comes out, another cell should be active, which is Yes, oxyntic cell, that is which secretes H plus Cl minus, right? Now, why? Because important point you need to understand is this: when pepsinogen comes here, there should be a pool of HCl. Now, first thing you need to understand is this: whenever at first pepsinogen comes out, more and more pepsinogen comes out at random phases. That means whenever chief cells start discharging the pepsinogen, now listen, whenever chief cells start discharging the pepsinogen, you need to understand right now is this, that whenever pepsinogen is thrown out of the chief cells, then at least more and more pepsinogens are coming for the first time. Why? Because pepsinogens were already pre-produced in the chief cells, which were stored. Now they will fall here in the acid content. Now important point you need to understand is this, that before the secretion of this chief cell, secretion of oxyntic cells must start by the help of acetylcholine receptors, that is M3 receptors. Now important point you need to understand is this, that whenever pepsinogen comes out and have a contact with this HCl, then what happens is this: pepsinogens are converted into pepsins. Now important point you need to understand is this: suppose this is your pepsinogen, okay? Suppose this is your pepsinogen. Now important point you need to understand is this, that this is 44 amino acid residues, okay? This is last 44 amino acid residues. Now important point you need to understand is this, that this amino acid here is susceptible to any pH change, That means whenever there is a increase in pH, this particular amino acid will be affected, and it will break the amino acid peptide bond. Okay, it will break the amino acid peptide bond, and these 44 amino acid residues are removed, and the remaining this portion, this whole was actually pepsinogen, and this remaining portion is actually the pepsin, the active enzyme. Clear? This remaining portion is actually the pepsin. Now important point you need to understand again here is this: pepsin has two very important characteristics, that is it's an endopeptidase, right? It's an endopeptidase. Another important point you need to understand about pepsin is this, that it's an aspartate protease. What do you mean by this statement? Now listen, suppose we have many proteins in the world. We have many proteins, still many proteins are not discovered yet. Now important point you need to understand is this: there are many big large proteins, but we have only 20 amino acids or 22 amino acids. Now what do you mean by this? That means each and every amino acid gets repeated many a times, right? Clear? Suppose you want to make a peptide of 500 amino acids, and you have only 21. That means what? You cannot make a unique peptide from unique amino acid sequences. Why? Because you cannot make any new amino acid. That means your amino acid sequence will contain the repetitive amino acid units, right? Now important point you need to understand here, is this: suppose this is your pepsin, okay? Suppose this is your one of the protein. Suppose this is your one of the protein. This is your N terminal, this is the protein C terminal, and this is another protein. This is N terminal and this is C terminal. Now I have already told that pepsin have two important characters. That is, one is it's an endopeptidase, right? It's an endopeptidase, and another is it's an aspartate protease. What do you mean by aspartate protease and endopeptidase? Now listen, let me give you an example, then you will understand both these statements. Now listen, this is N terminal, this is C terminal. Now suppose this is aspartic acid. Now So there are many, there are only 21 amino acids, okay, and there are many proteins. There are thousands and crores of proteins present in your in the world, not crores, but at least in lakhs. But important point you need to understand is this, that these proteins have many amino acid sequences, 5,000, 6,000, for example. Now 21 amino acids are there, and there are 5,000, 6,000 amino acids which constitutes a protein. Now important point you need to understand here is this, that means some amino acids are getting repeated. Now important point you need to understand here is this, that suppose due to this repetition, aspartic acid residue, or this D stands for aspartic acid, may get repeated many a times. Now important point you need to understand is this, suppose there is 3 aspartic acid residue here. Now suppose here is again 3 aspartic acid residue. This D stands for aspartic acid. Go for amino acid, biochemistry class, you will understand what is, what are the short signature of these amino acids. Okay. Now why it is called D and why phenylalanine is F. Now important point you need to understand is this, that suppose you are actually instructing this pepsin to cut this protein. This is suppose protein number 1, this is suppose protein number 1, and this is suppose protein number 2. Suppose you are telling this protein, suppose you are instructing this pepsin to convert this or to digest this protein. Now important point you need to understand is this, you will get 3 sections here. You will get 3 small peptides. Now here in the second protein, what you will get is 2 sections. Why? Because they are endopeptides. Therefore they will not work from the terminal. Because they are endopeptides. Okay. Now important point you need to understand here is this, that in this case you will get what? How many? Sorry, you will get here 4, right? 1, 2, 3, 4. But here you will get 1, 2, 3 sections, right? Okay. So now Now important point again you need to understand, suppose there is N terminal, there is C terminal, there is in the first only there is D, in the end there is D, and there is D in the middle. Then how many sections you will get after pepsin digestion? You will get 2 sections, okay? So this is the importance of these two, points, that is it's an endopeptidase, that means it only cuts inside peptide bonds. And aspartate protease, what do you mean by aspartate protease? Aspartate here represents the affinity of that particular enzyme. That means that enzyme actually finds out the aspartate and cut the aspartate adjoining peptide bonds. Okay. Now important point you need to understand again. Suppose this is your N terminal and this is your C terminal, and there is D and D here. Now my question is this, from where pepsin will act? From where pepsin will start their action? From N terminal or from C terminal? Where? They will always start their action from N terminal. Clear? They will always start their action from N terminal. Clear? Okay. Now important point you need to understand is this, the favorable pH for pepsin action. The favorable pH for pepsin action is 1.2 to 2. Okay, 1.2 to 2. And another thing you need to understand is this, pepsin is inactivated at 6.5 pH. Clear? Is inactivated at 6.5 pH. Okay. Okay. Now, so we now know why pepsin is called endopeptidase, why it is called exopeptidase. Now we will talk about its secretion. Now important part, or regulation of secretion. Suppose this is a pepsinogen secreting cell, that is chief cell. This is a chief cell. Okay. Now first point you need to understand is this, that suppose this is a vesicle containing pepsinogen. Now first point you need to understand is this: any GI tract secretion is increased due to parasympathetic activity. Okay, any GI tract secretion is increased due to parasympathetic activity. Now important point you need to understand is this: whenever food enters the stomach, okay, now important point you need to understand is this: whenever food enters the stomach, this leads to a vagovagal reflex action. This leads to a vagovagal reflex action, which ultimately leads to secretion of pepsinogen, and this pepsinogen is getting affected by HCl, and HCl converts this pepsinogen into pepsin by disrupting a peptide bond which releases 44 amino acid residues, right? Now important point you need to understand is this, that when food enters, then brain gets excited. Okay, now when food enters, this information that food is entering is carried by a vagus nerve. Okay, it's carried by a vagus nerve. Now important point you need to understand here is this, that the motor system in this case is also a vagus nerve. Okay, now as the sensory system as well as the motor system is a vagus nerve, that's why it's called vagovagal reflex. Now important point you need to understand is this: suppose this is suppose food activates this motor vagus system. Now important point you need to understand is this: from here acetylcholine will be secreted, and acetylcholine will now bind with M3, that is muscarinic receptor type 3 receptors, and they are coupled to GQ, and this leads to increase in intracellular calcium concentration. If you don't understand, try to learn the chapter of cell signaling. Now, so increase in calcium concentration leads to what? CAM kinase activation, and CAM kinase activation leads to, now listen, how these vesicles move towards the cell membrane so that they can be ejected out or they can be exocytosed. Now important point you need to understand is this: for exocytosis of this particular vesicle, you need to understand a very basic thing, that is, for exocytosis of this particular vesicle, the proteins which are present here should be phosphorylated. Okay, which are present in this actin myosin pathways should be phosphorylated. These actin myosin pathways, where the vesicles rest, they are like railway roads, and on these railway roads trains are suppose standing. To start the train, you need electricity, right? Now important point: to start this train you need phosphorylation of these proteins. Okay, now these protein phosphorylation leads to exocytosis of this content, pepsinogen, and pepsinogen is right now released into the pepsinogen then is released into the stomach and is acted upon by HCl, then it is converted into pepsin. Okay, now important point you need to understand here is this, that there are also other machineries or mechanisms. Now, if you think that adrenergic system actually inhibits this total activation of pepsinogen, you are wrong. Why? Because there is beta 3 adrenergic system which is coupled to GS, and it leads to cyclic AMP concentration increase in cyclic AMP concentration, and this cyclic AMP concentration increment leads to protein kinase activation, and this leads to phosphorylation of these proteins which are actually controlling the exocytosis of pepsinogen containing vesicles. Now, important point to understand is this, that a very funny thing actually occurs. That is, when suppose this is gastric pit. Okay, suppose this is gastric pit. Suppose this is your chief cell. When chief cell secretes pepsinogen and they are converted into pepsin, pepsin acts on proteins, right? Pepsin acts on proteins and converts them into peptides, right? Pepsin converts them into peptides, right? Now, important point to understand is this: pepsin is converting proteins into peptides. Now, important point to understand is this: now, suppose this is your chief cell, pepsinogen has been secreted, pepsinogen is getting converted into pepsin by the help of HCl, and pepsin acts on proteins and is converted into peptides, and these peptides actually help in further secretion of pepsinogen. How? This is very important. Now, important point to understand is this peptide which is generated by pepsin action. This peptide actually goes into the bloodstream and activates a cell called G cells. Okay, G cell is gastrin secreting cell, and they secrete gastrin, and gastrin is a hormone which actually helps in HCl secretion, right? And this now listen, HCl is actually secreted by oxyntic cells, and chief of peptic cells secrete pepsinogen. Now, gastrin release. Now, these cells secrete gastrin. Now, gastrin activates oxyntic cells so that they can secrete HCl, and now HCl again acts on more and more pepsinogen to convert pepsinogen into pepsin, and active pepsin convert proteins into peptides. Okay, now two important point you need to understand is this: what is the difference between peptide and peptone? Now, what is peptone? What is proteose? These are very important point. Now listen, peptones and proteose. Proteose is never produced in your body. Proteoses are never produced in your body. Peptones can be produced in your body. Now listen, what is the basic difference between peptones and proteoses? Now listen, they are actually not biological terms. These peptones and proteoses are actually not biological terms. They are actually physiological terms, or specifically biochemical terms, not physiological, biochemical terms. That is, if any portion of the protein is precipitated by the help of NH2SO4 whole twice, that is ammonium sulfate, NH2SO4, if any, not NH2SO4 whole twice, sorry, if any product of protein digestion is precipitated by ammonium sulfate, then it is called peptone. And if any supposed digestive product is not precipitated using NH4 whole 2SO4, then it was called proteose. Now, important point you need to understand is this: proteoses are only required in chemical laboratory for specific tests of proteins, and peptones are also required for only small tests of proteins like biuret test and all these things. We don't in the body these proteins and peptones are produced really in small amounts, but in most of the cases peptides are more important. Clear? Now important point you need to understand is this, some activity or motility of stomach also helps in secretion of pepsinogen and HCl. Okay. Now a very interesting point you need to understand is this, that prostaglandin E2 via a receptor can lead to activation of this pepsinogen release. Okay, prostaglandin E2, okay, by the help of a receptor known by the name of EP2. Clear? Okay. So this was your digestion of proteins. Now, okay, so what is the just before ending the digestion of proteins in stomach, you should understand that what is the end product of digestion in stomach. The end product is peptides, not amino acids. Okay, clear? Now we will talk about digestion of peptides in the small intestine. Clear? Okay, so now we will be talking about digestion in small intestine. Okay, now we will be talking about digestion in small intestine. Now important point you need to understand here is this, that when we are talking about digestion in small intestine, you need to understand that mainly mainly one organ and one particular part of the organ is actually helping us. That is, small intestine itself is helping us. Small intestine itself is helping us, and pancreas. Now, I have already told that those persons who is having gastrectomy, they actually don't have any problem. That is, their total on an average nitrogen balance in the body is okay. Now those having pancreatitis, they have severe problems in digestion. They have bulky stool. Okay. Now why this occurs is, now when we are talking about the digestion in small intestine, we need to understand that suppose this is your pancreas. Okay. Okay. Now suppose this is pancreatic duct, this is your pancreatic acini. Pancreatic acini move inside the acini. Pancreatic duct move inside the acini. Now suppose this is your acinar cell. Suppose this is one acinar cell. Okay. Now important point you need to understand is this, that this acinar cell is activated by vagus nerve. Okay. Now listen, I have already told that every secretion of GI tract is actually positively affected by vagus nerve, that is parasympathetic activity. Every secretion is increased due to parasympathetic activity. Now listen, due to this what happens is, when acinar cells becomes activated by vagus nerve, then they start secreting pepsinogen. Then they start secreting, then they start secreting trypsinogen. Okay. Then they start secreting trypsinogen. Now important point you need to understand right now is this, that whenever trypsinogen is coming out of the acinar cell, then they are active. Right. Now suppose there is a blockage. Now listen, suppose trypsinogen is coming here and suppose there is a blockage here. Now what will happen? Trypsinogen will be auto activated, and this will lead to chaos there. That means all these tissues will be digested. right? This is called, when these type of things are occurring here, this is known by the name of pancreatitis. Pancreatitis. Okay, this is known by the name of pancreatitis. Okay, now listen with all your attention. So suppose this is a healthy pancreas. Suppose this is your pancreas and it is healthy, I guess. Now it is coming here. Now this is your C-shaped duodenum. Okay, this is suppose your duodenum. Okay, clear? This is suppose your duodenum. Now important point you need to understand is this. Suppose this is your duodenal cell. Suppose this is your duodenal cell. Okay, now very important point you need to understand here is this, that these duodenal cells contain an enzyme here, contains an enzyme called enterokinase. This is called enterokinase. Okay, enterokinase. That means they are present, these enterokinases are actually present in the lumen of duodenal cells. Now important point you need to understand is this, that enterokinases are also present in the jejunal cells. Jejunal cells. Clear? Now important point you need to understand is this, that when enterokinase comes in contact, or when trypsinogen comes in contact with enterokinase, they are converted into the most important enzyme of all in the digestion of protein, that is trypsin. Trypsinogen is converted into trypsin by enterokinase, okay, which is present in the lumen of duodenal or jejunal cell. Now important point you need to understand here right now is this, that when trypsin comes in contact, trypsin actually, from acinar cells, not only trypsinogen is coming. There is chymotrypsinogen, there is carboxypeptidase A, there is carboxypeptidase B. We will talk about that, don't worry. Now all these enzymes are actually activated by trypsin. Now in short we will introduce the trypsin. Now what is trypsin? Now listen with all your attention possible. Trypsin is a serine protease. Now you know what is serine protease. Like pepsin is an aspartate protease, it's a serine protease. Okay, that means it will, whenever they will see serine, they will try to cut there. It's a serine protease. Now again important point you need to understand is this, trypsin is an endopeptidase. Can I make your work a little more easier by showing which of the pancreatic enzymes are endopeptidase and exopeptidase, right? Now listen, okay. Enterokinase is an endopeptidase, okay? Endopeptidase. Okay, now otherwise you wouldn't have got any protein, you would have got amino acids. Now listen, enzymes which are coming out of the pancreas, the enzymes which are coming out of the pancreas, they have been classified into two categories, that is exopeptidase and endopeptidase. Now endopeptidases, now listen with all your attention. Trypsin, chitri, carboxypeptidase A, carboxypeptidase B, okay, carboxypeptidase peptides A and B, all are endopeptides. Clear? Now, what is the enzyme which is associated? Sorry. Carbopeptides A and B are exopeptides. Carboxypeptides A and carboxypeptides B. Now, elastase is an enzyme which is activated again by trypsin. Now, what is the similarity between trypsin and all these enzymes? Now, the similarity is these enzymes are all activated by trypsin. They are all activated by trypsin. Clear? By proteolytic cleavage. Okay? Now, you need to understand that it's an endopeptidase. Now, very important point here you need to understand is this, that what is the pH at which pepsin works? The pH at which pepsin works is around 7.5 to 8.5. 7.5 to around 8.5. Okay? 7.5 to around 8.5 trypsin works. Now, important point here you need to understand is this, that trypsin, when it is getting activated, it activates actually chymotrypsin. And another thing, when trypsin gets activated, very important point you need to understand is this: when trypsin gets activated, it auto activates trypsinogen to trypsin. That means trypsinogen is getting converted into trypsin by the help of enterokinase, right? Now, trypsin acts on trypsinogen to create more and more trypsinogen. It's an autocatalytic activity. Okay? It's an autocatalytic activity. Trypsin shows autocatalytic activity. Clear? Trypsin shows autocatalytic activity. Now, so important point you need to understand is this. So, how trypsin actually works? Now listen, it's an endopeptidase. That means it will create what? Peptides. It will create peptides, right? It will create peptides. Now, important point you need to understand is this, that chymotrypsin actually is what? Chymotrypsin again is a endopeptidase, right? That means chymotrypsin, trypsin, elastase, whatever we talk about these things, how much we talk about these things, the first point you need to understand is this, that they are all endopeptides, and neither trypsin nor chymotrypsin or elastase, they will never give you any amino acid. Understand this very basic thing, okay? They will never give you any amino acid. Now, which enzyme will give you the amino acid for absorption? Carboxypeptidase A and B. Clear? Carboxypeptidase A and B will give you the amino acid for absorption, but they have certain conditions. What are these? Now listen. Why they will give amino acid? Because they are exopeptides, right? Okay. Now listen. A general rule about protein digestion in small intestine is, general rule about this is, you need to understand that suppose from stomach large peptides are coming to the small intestine. Now these large peptides are acted upon by two different enzymes, okay? These large peptides are acted upon by two different enzymes in the two different mechanism. Number one is chymotrypsin plus elastase. Number two is trypsin. Okay, now listen with all your attention. Suppose this is peptide. This is N terminal and this is C terminal. Now important point you need to understand is, suppose this is a peptide. Now important point you need to understand here is this, that suppose I am telling you now, after digestion with chymotrypsin and trypsin, this is we have got. Now listen, suppose this is a large peptide, and after treatment with trypsin plus chymotrypsin, we have got segments. We have got suppose 2 segments or 3 segments, whatever it is. Now how will you determine that which segment is actually the product of trypsin digestion and which segment is the product of chymotrypsin digestion? It's very easy. Now listen, any smaller peptides which is obtained from these larger peptides will be obtained from trypsin digestion when the peptide ends with a C terminal basic amino acid. Suppose this is there are 1, 2, 3, 4, 5, 6. Now listen, suppose trypsin clips off suppose 3 amino acids. Now how will you understand that trypsin has clipped this particular part? You will understand by checking the C terminal. Okay, if you want to know whether a smaller peptide has been created by trypsin or chymotrypsin, you will have to check the C peptide or C terminal of the peptide. Okay, now listen, if the C terminal of the peptide contains a basic amino acid, then no doubt this cleavage was done by a trypsin molecule. Clear? Now suppose this is the molecule, suppose, which has been obtained from this. Now listen, I am not telling what is the sequence of these, first amino acid, second amino acid, third amino acid. Why? Because you need to understand is this, that this is a, this is an arbitrary, sequence of amino acids. Now, suppose you have got this. So where you will check that whether it is a trypsin activated molecule or chymotrypsin activated or chymotrypsin cleaved molecule, you will have to check the C terminal. Now, if you get a neutral amino acid in the C terminal, no doubt, if you get a neutral amino acid in the C terminal, there is no doubt that this is done by chymotrypsin. This cleavage is done by a chymotrypsin molecule. Clear? Now what will happen is, suppose both of them are acted upon by carboxypeptidases. Okay, both of them are acted upon by carboxypeptidases. Now what will happen is this, suppose if both of them are acted upon by carboxypeptidase A and carboxypeptidase B, suppose, okay, suppose B. Now suppose they are acted upon by carboxypeptidase A or B. Now what will happen is this, carboxypeptidase is what? An exopeptidase. That means what? They will cleave these amino acids and they will create what? Single amino acids. That means they will cleave the peptide sequence and they will try to create single amino acids. Why? Because they are exopeptidase. Clear? So now we will be studying how these single amino acids are getting absorbed in your body. Okay? Okay.